Hello everyone, I welcome you back to another important topical past paper practice. For this chapter that you have for your IGCSE Biology, and this goes with the name of enzymes. But before I proceed, I would like to mention that all of the questions that I have included from the year 2010 to year 2015, over there your theory paper had a different code and that was 3.1, 3.2 and 3.3. And from 2016 coming till 2025, your theory paper variant would be 4. So no matter if this is 3 or this is 4, the questions that I have taken, they apply to your current syllabus pattern. Now, they have asked in the year 2010, this was October, November, the variant was 3.2, and here we had this question number 1 with us. And this says that we are showing you figure 1.2, and this is a transverse section through a buttercup root at the end of the cold winter W, and then at the end of the warm, moist summer that we have with us. Now, if you see here, we have got this W, we have got this S. The W shows you a cross section of the buttercup root, and that is actually at the end of the winter. And this S is at the end of the summer that we have with us. Now, what they have to say is that at the end of the winter, these cells contain very few starch grains. So, if you just look on W, that shows the end of the winter. And they are saying that the starch grains that are present within the buttercup root, they are very few. But then they say that at the end of the summer, most of the root cells contains many starch grains. So here this S we have got it for the summer, and if I were to just zoom in, you see a lot of deposition of the starch here. And if we just look on toward the part that they gave from here, this was for 3 mark, and they asked to describe how the enzymes in the root cells they synthesize starch. Now, if I were to recall, what we know is that this starch is a complex sugar, and whenever we are required to be forming it, so I need to combine many glucose with the help of a condensation reaction. And for that purpose, these glucose molecules they need to be fixing into the active site of an enzyme. And when they would be linked up together with the help of a bond, and as a result of that, we would make the starch out of it. So since we have to write it in context of the enzymes, so I would say that the enzyme they had an active site, and that active site has a shape complementary to be binding with the glucose. So the glucose would bind with the active site, and when the glucose would come into the active site of the enzyme, so these glucose molecules they would be linked up with the help of the bond. And by that, these glucose molecules, they would keep on linking by the condensation reaction. And as a result of that, we would end up making the starch molecule. If I were to move further to the part D, here they say is that as the temperature is increased, for example, from 10 degrees centigrade to 30 degrees centigrade, enzymes activity increases. So you see that the temperature, when this is elevated from 10 to 30 degrees centigrade, so we are increasing the temperature. And when the temperature is increased, so that basically indicates is that the kinetic energy of the enzyme molecule and that of the substrate it would increase. And when their kinetic energy would be increasing, though so both of these they would start to move more randomly. And when they have started to move more randomly, so the frequency of the collision between this enzyme molecule and the substrate molecule they would increase, and that would help in making more of the enzyme substrate complex. So for 2 marks, they are asking that explain how increasing the temperature affects the enzymatic activity. So what you were required to be writing was that the increase in the temperature, this would enhance the kinetic energy or the movement energy of the enzyme and the substrate molecule, and the frequency of the collision between the enzyme and the substrate it would increase, and we would make more enzyme substrate complexes out of it. If I were to go to this second part, this is one of the frequently asking question, and this arrived in October, November of 2011, and this was question number 4 with us. Now, if we just simply see what the question has to say, that an investigation was carried out to determine the effect of different temperatures on the activity of the thrombin, and they are showing you the results below. Now, if I were to talk about the thrombin enzyme, what we know as that this thrombin enzyme this basically converts the soluble protein fibrinogen that we have in the blood plasma, and it would be converted into the fibrin, and that fibrin it would make the meshwork, and that meshwork would trap the white blood cells and the platelets, and ultimately we end up making clot out of it. Now, since we are changing the temperatures for the enzyme given, so this temperature would be acting as the independent variable. Why we are saying this is an independent variable? Because in each of the investigation, a different temperature would be provided to the enzyme. And now they would see that how changing this temperature would have an impact on the activity of the thrombin enzyme. Now, if you just look on toward the graph, on the y-axis, they have given you the activity of the thrombin. They did not specify the units. They have simply written it generally that these are the arbitrary unit. And if we go on toward the x-axis, then we have the temperature in terms of centigrade. If I were to describe this graph, so what you see is that when the temperature is increased from 0 to 40 degrees centigrade, so the enzymatic activity of the thrombin is increasing. Another valid point is that if you were to see the gradient, so between 30 and 40 degrees centigrade, the gradient is more steeper. So this means that in between these two temperatures, the enzymatic activity is increasing at a faster rate. But if I just talk about 0 to 30 degrees centigrade, the activity of the thrombin enzyme is increasing, but the gradient here for the graph is not as much steeper as this point of the graph. So here the activity of the enzyme is increasing but at a slower rate. Now if we just indicate with respect to 40 degrees centigrade temperature, so this 40 is the optimum temperature of the enzyme activity because here the enzyme is exhibiting the maximum catalytic activity of 8 arbitrary unit. And if I were to go beyond, so from 40 degrees centigrade to 50 degrees centigrade, we see a very steep decrease in the enzymatic activity. And the reason is that when the temperature is increased beyond 40 degrees centigrade, so the active sites of the enzyme they start to lose their shape. And when the active site would start to lose the shape, so the substrate binding with the enzyme molecule it decreases. But if I just see from 50 to 60 and then 70, so here we see again a decrease in the catalytic activity of the thrombin enzyme, but this is not as much steep as compared to the temperature we have got it from 40 to 50 degrees centigrade. Now, if we just look at 70, the graph is touching the x-axis. So at this point, the enzyme is completely denatured. And if we just simply see at a 0 degree centigrade temperature, again, enzymatic activity is 0. So at a very low temperature of 0, the enzyme molecule are not moving. So they would not bind with the substrate. And that is why the activity that we see here at 0 degree centigrade is 0. But at 70, the enzyme molecules are getting denatured. So if we were to move further, here they have given you total of 3 marks and they have asked that explain why thrombin functions slowly at 5 and does not function at 70. If I were to take you back toward the graph here, we have got the value of 5 degrees centigrade. And against this enzymatic activity that we see is this is 0.5 arbitrary unit. So why this so happened that at 5 degrees centigrade, the enzyme is working slowly. The reason is the temperature is low. So there would be fewer collisions that would be happening between the enzyme and the substrate molecule. So that is why the enzymatic activity is slow here. But if we refer to the explanation that we are required to give at 70, so you see 70 is such a high temperature. And at this temperature, the enzyme would have lost the shape of its active site. The active site has been deformed or the enzyme molecule has got denatured and it no longer remains complementary to the shape of the substrate. And that is why we have got zero catalytic activity at such a high temperature of 70 degrees centigrade. So how you were required to be writing the answer as such? So you would say that at 70 the enzyme is completely denatured and the shape of the enzyme's active site is lost, and that is why it would no longer bind with the substrate molecule, we say. Moving onward, for 1 mark, they are saying that suggests how the activity of the thrombin was determined. And as I have indicated earlier, that this thrombin enzyme have an ability to convert the soluble fibrinogen to the fibrin. So if I were to measure or determine that how the activity of the thrombin is being affected by the temperature. So I would say that I simply would count the time taken for the fibrin to form. Like if more of the fibrinogen would be converted into fibrin, so less time would be taken for it. But if the activity of the thrombin would be slow, so more time would be taken for the fibrin to form. So this is how we were required to be um, telling our examiner that how the activity of the thrombin would be determined. Now here they ask for the total of 2 marks that we are required to say two conditions that would have been kept constant during the investigation. And you see that on toward the x-axis here, the temperature is shown. So anything that comes on toward the x-axis, this is your independent variable. And what I know from my prior knowledge is that the activity of the enzyme is impacted not only by the temperature, but also by the pH, by amount or concentration of substrate, by the amount and the concentration of the enzyme. So I need to keep the others constant because if I would not keep them constant, I would not be able to see the effect of the temperature alone. So we would say to your examiner that since you are changing the temperature, which is your independent variable, so you have to keep the pH, or you could write amount or concentration of substrate. And if you just forget about the substrate thing, so you could say that we are required to be keeping the amount or the concentration of the enzyme constant. And in that way, we would be effectively measuring that how come the temperature is impacting the enzymatic activity of this thrombin enzyme. Moving onward to this question number 3, and this was for the total of 4 marks, and this appeared in May June of 2012. And they say the action of the enzyme is often explained in terms of lock and key model as shown in figure 3.2. So, what the log and the key hypothesis actually suggests is that this enzyme molecule that is shown to us here in the figure 3.2, so the enzyme would always act as a lock, and the substrate that we have got, this would act as a key. And if we were to simply see that this enzyme has got the shape of the active site and this is complementary to the shape of the substrate. So the substrate is binding with the enzyme's active site. So we are making this complex out here shown in the middle, and this is the enzyme substrate complex. And later you see that the enzyme substrate complex would be converted and the enzyme would remain intact. And the product that we have formed out of the substrate that are shown here, so they are leaving the active site of the enzyme. So they have to ask it for the total of 4 mark, as I've said, that we are required to use information that is shown in figure 3.2, and we need to explain that how the enzymes they work to break down the nutrient material such as starch. So since I have to be explaining the enzymatic activity, and you see at the top, they themselves has indicated that this is lock and key hypothesis. So we were required to be writing in this context, and this figure is again a clear hint that how you were required to be writing down the answer. So as I have explained it above, we would say that the shape of the substrate, which is actually starch, so this is complementary to the enzyme's active site, like I have written out this point explanation. And in doing so, the substrate molecule, this would act as a lock and the enzyme would act as a key. And now since this is a situation, so the substrate would bind with the enzyme's active site, and as a result of that, we are making an enzyme substrate complex out of this. And later, the substrate which is starch, so this would be converted into its respective product, which would be glucose with us. And now this is leaving the enzyme's active site so that the enzyme can bind with another substrate molecule. So this is how you would have scored total of 4 marks for the explanation that you were required to provide with context to figure 3.2, which they gave you in the exam. If I were to move further, they say that we have got the enzymes, and these enzymes they are available in the bean seeds, and they get activated when there is this seed germination. Now you know that when the seeds are germinating, so a lot of enzymes would be activated, and those enzymes they would break down the stored starch, the stored protein, and the stored fat into their constituent molecules, and that would be used by the seedling or the embryo, and it would grow into a mature plant. They say some of these enzymes break down protein which is stored in the seeds. Now, If I have got the protein stored in the seed, so protease enzyme has to be released by the embryo within the seed, and the protein would be broken down into amino acid. Now, what they have to say further is that a large number of the bean seeds were soaked and germinated. Now, why are we soaking the bean seeds? Because in water, because that would activate the enzymes in the embryo and the seed would start to germinate. Now, further they say is that the researcher took samples of the germinating seed over a period of 15 days, like whatsoever the seeds which they soaked and they were germinating. So the scientists they are collecting samples of the germinating seed and for the total of 15 days. Later, the seeds were chopped into small pieces and they were crushed with the water to make an extract. So after they have collected the samples for 15 days, now they are chopping them up. And for chopping, they're adding water along with it, and this is making an extract. Now, what they have to say further is that equal quantities of the extracts were placed into a protein solutions, and the pH of the protein solution they were kept at pH 5 and pH 8. So you see that for the protein solutions we are changing the pH, so the pH would be acting as an independent variable here. Further, they say that the activity of the enzyme in each extract was determined by recording how quickly the protein was broken down. So you see, if I were to just summarize this, we had got these bean seeds. When they germinate, they produce an enzyme, and that enzyme breaks down the protein. The scientists they soaked and then they germinated these bean seeds for the total of 15 days, and later they chopped them up in the water and they have made the extract out of it. Now later they took the two test tubes. One of the test tubes and the second test tubes they have the protein solution, but their pH was maintained at a different value. Like the first protein solution, this was maintained at pH 5, the second at pH 8, and now they are adding the seed extract to both of the protein solutions. So we know that this seed extract that has come all the way from the bean seeds, so this has a protease enzyme in it, and they are just trying to see that how come the protease enzyme that we have in the seed, they would break down the protein solution that we have got in both of these test tubes. If I were to take you toward the graph, so you see is that on toward the x-axis we are given the time in term of days, and on toward the y-axis they have given you the enzymatic activity and that too in the arbitrary unit. Now, if you just look on toward the graph, so we have got two lines. The dotted line is basically indicating the enzymatic activity in the test tube in which the pH was maintained at 8, and the darker line that we have got over here. So this is indicating the enzymatic activity that of from the seeds into the protein solution that was kept at pH 5. Now, if I just talk about for the protein solution that was kept at pH 5, so you see that the activity of the enzyme that is shown here with the help of a darker line. So this gradually increases till day 3, then this falls till day 5, and then from day 5 and until coming here till day 11 that we are having here, because you see that in between these two values we have got total of 2 units. So the half of this is representing like 1 day. So here we have got 11 days. So from day 5 till day 11, the enzymatic activity is steeply increasing. But then again from day 11 till day 15 that we have here, the enzymatic activity in the solution that was kept at pH 5, this is steeply decreasing. But if I were to talk about the protein solution that was kept at pH 8, so the protease enzyme in that, so this is indicated by the dotted line, and this increases as a day passes. And then at some days, like if we just look here, from these days from 5 to 7 days, so the enzymatic activity is little bit constant. And here you see this is increasing, but here there is a very gradual decrease, or you could say that here the enzymatic activity is a little bit constant. If we were to move onward, they have to ask it for the total of 3 marks, that we are required to describe the activity of the enzyme in the extract that was kept at pH 5, and we have to say it for over total of 15 days. Now wherever the word describe comes, so we just simply have to quote him the values that come on toward the x-axis and y-axis. Now we simply have to crack some points in terms of description for the enzyme solution that was kept at a pH of 5. So you see it, like I am just going to zoom it more. So I would say that from day 0 to day 3, there is a gradual increase in the enzymatic activity, and we have attained the enzymatic activity of only 2 arbitrary unit, because we have got like marking from 0 to 5, so every small division is representative of 1 arbitrary unit. Then later we would say that the protein solution that was kept at pH 5 from day 5 till day, you could say 11, the enzymatic activity is increasing. And how much this is increasing? From the total of, you could say, 1 arbitrary unit that we have here, and max this is coming till 37 arbitrary unit. And another description point that we have got over here is that from day 11 that we have here until day 15 that we have here, so here the enzymatic activity is decreasing, and then you require to quote him that this decreases from 37 arbitrary unit, and ultimately this comes to 14 arbitrary units. So these are the description. We are not required to give him any of the explanation. Now, what the examiner has done for the part of this question that applies to this part specifically, and for 3 marks, they are asking that you are required to describe the activity of the enzyme in the extract at pH 5 over 15 days. And as I have mentioned earlier, what we would say that this enzyme would show very little activity of 2 arbitrary unit till day 3. Now you see like here we have got day 3, and from day 0 to day 3 the enzymatic activity is very little or very less till day 3. And then further I have said that the enzyme activity, this is increasing steeply from where? Day 5 from here, and then ultimately coming till day 11. And here we have got the maximum, you could say, a enzymatic activity of total of 37 arbitrary unit. And then afterward, we would say that the enzyme activity, it decreases from day 11 that we have here till day 15. And by that, I have like simply said that this is reduced by 23 arbitrary unit. Now I have taken the difference actually. Like here we have 37, and here we have got like a value of 14. So I have subtracted this 14 out of the 37 to just simply tell him that from day 11 till day 15, the decrease in the enzymatic activity in terms of the arbitrary unit is 23. So this is how you would score 3 marks in terms of the description scenario. Now moving onward, for the total of 3 marks, in the part E, they asked that the researchers concluded that the beans contained 2 different enzymes that break down protein, and we are required to be stating the evidence from figure 3.3 for this conclusion. Now, the idea is very simple, that the enzymes are different because at the provided pH of 8 and the pH 5, the shape of the graph that we are given, this is different. So this means that the mode of activity of the enzyme at both the pH is different. The other idea is that the enzyme solution that I kept at pH 8, so this has got an optimum catalytic activity, and it was being exhibited at a later day of 15. But if we talk about for the pH 5, so this was achieved at the initial days, or the enzymatic activity was optimum in the earlier day, or it was attained fast up till day 11. And other than that, we would see that the enzymes are very specific to the pH, because for the first, you could say seed extract that I kept at pH 5, so you see that it is increasing steeply between day, you could say 5 to going till day 11. So this would mean that the enzyme here is actually preferring the acidic pH. But after day 15, the enzymatic activity is no longer shown. So this means that the enzyme has stopped working after day 15. So how you were required to be writing the answer here, we would say that the shape of the lines that we see at pH 5 and pH 8 is different. And the idea is that the line of the graph is a clear indication that they are not only working at the both pH, but they are also pH specific. And the enzyme at pH 5, this was maximum by the day 11, as I've indicated above. And the other enzyme that was kept at pH 8 from the seed, so this has got the maximum activity by day 15. And they both enzyme, by the lines we would say that they prefer to work in the different pH. So this is how you would score total of 3 marks here. If I were to move beyond, so for this question number 4 that appeared in October, November of 2012, And here they have asked that enzymes are biological catalysts, and we are required to be defining the term catalyst for this. And the total marks given are 2. Now, you see is that whenever we have to be defining the word catalyst, so we would say that this is a chemical substance, and what this chemical substance would do, that it would increase the rate of the chemical reaction, and by doing so, it would itself not be chemically changed or the modified. If they had asked here to define the term enzyme, so we would simply say that the enzymes they are the biological catalyst, the other point would be same. If we were to be coming toward the second part, and here this is an investigation which they have presented. What they say is that urease is an enzyme and that is found in bacteria and in the seeds of some species of the bean. Like simply, either we have the bacteria or we have some species of the beans and they have this enzyme in this, and this is the urease enzyme. And they say that the enzyme catalyzes a reaction. How? Like when the urea in the presence of water, when this would be acted upon by the urease enzyme that we have in the bacteria and that we have in the species of the bean. So this urea would be broken down into carbon dioxide and the ammonia gas. Further, they say is that when the production of the ammonia is increased, so the pH of the area around the bacteria, like ammonia is basically a kind of an alkaline gas. And when this is released, so the area that we have got surrounding the bacteria, the pH of that area would increase. Why? Because greater the pH value, so more alkaline the pH is. They say the formation of the ammonia can be used to study the progress of the reaction by testing the pH of the surrounding medium with a pH indicator such as the universal indicator. Like if I were to just simply draw a petri dish, and in the petri dish we have got this bacteria, and in the, you could say, surrounding media of the bacteria, what we have provided, this is the urea. So when the enzyme urease would be released out of the bacteria, so this urea, since if this is being broken down into the ammonia, so you see that here in the surrounding area, the pH, we can measure it with the help of the universal indicator solution. If the universal indicator solution would give us an alkaline value in term of the color, so we would say that the urease enzyme from the bacteria was helpful in breaking down the urea. Now, further, they say is that some students they carried out an investigation to find out if there was urease in the seeds of 4 different species of the bean. Like here, you notice is that the entire situation that was presented above, this was in context of the bacteria. But the statement which they have given over here, so here the scenario they have changed it. That the students now they have got 4 different species of the bean, and out of the bean they are of the view that the urease enzyme would be released out, and that by some mean or the other can break down the urea. If we were to move further, they say the germinating seeds were grounded up in the water and filtered to give an extract containing the proteins. So simply like I would take the germinating seed, I would crush them up in the presence of water, then I would filter the resulting solution and I would be making an extract, and that extract they say would contain the proteins in them. Now they say that each extract was added to the urea solution and the temperature of that was kept at 30 degrees centigrade for 30 minutes. Like I would have total of 4 test tube, like this is test tube 1, then I have test tube 2, then we have this test tube 3, and then we have the test tube 4. And in all of these test tubes, what we have added is the urea solution and the seed extract I am adding into all of them, and the temperature we are keeping it constant at 30 degrees centigrade, and the investigation was carried for the total of 30 minutes. Further, they say that we have included 2 of the more test tubes, they were 5 and 6, and they were also, you could say, included in the investigations. Later, when the experiment was run, so the scientists they were taking the samples from the reaction mixture at total of 5-minute interval, and then they try to test them with the universal indicator solution. So simply we have got total of 6 test tubes. Like we have the urea solution and then the seed extract is being added, and we will see that how come the urea in these test tubes they would be broken down. Now they have given you in the table the test tubes, and this is one, two, three, four, five, and six. And we have got these bean species with us, and they are soya, mung, jack, broad, and again, we have got the soya. Now this is being repeated. And then we have got no beans. So if I am adding no beans in the test tube 6, so this would be acting as a control experiment so that we can make the comparison with the rest of the test tubes. Now, if we were to talk about the urea solution, so they added the urea solution to all the test tubes, but except for the fifth test tube. And then in terms of the water, they have added them into just test tube 5 and 6, but none of the water was added to the first four test tubes that we have here. And now they are giving you the values in terms of presence of alkaline pH at interval of 5 minutes. So if I just simply talk about the first test tube, so for the first 15 minutes, none of the urea was broken down, and since no ammonia would be produced, so the universal indicator value is presented with a cross. But for the 20, 25, and 30 minutes, the universal indicator gave the color in terms of the alkaline value. So what I can conclude here that this soya bean species, this has a urease enzyme, and that is why we have got the positive test in terms of the alkaline pH with the help of the universal indicator. Now, if I were to talk about the mung beans, so they do not have the urease enzyme, it would not break down the urea into ammonia. So at any of the given time, we would not get any of the alkaline pH. If I were to talk about the jack, so again we have got the ticks here for the minute 15, 20, 25, and 30. So this again has the urease enzyme in it. The broad bean does not have the urease enzyme. The soya, now since we haven't added any of the urea solution into it, like here soya, when this was given urea, so we have got positive result. But when we have soya and we do not provide the urea substrate to it, so again we are not getting any of the alkaline values. And likewise, if we have no beans, so there would be no urease enzyme, and ultimately again, we would not have any of the positive result here. And they have given you the key as well, that tick mean the alkaline pH and no tick mean that there was no alkaline pH. Now, what they have to be saying is that we are required to explain that why the test tubes were kept at 30 degrees centigrade and the marks were 2. So the idea is very simple, that the activity of the enzyme is impacted by the temperature, by the pH, by the amount of the substrate, and by the amount of the enzyme. So the temperature is important with respect to that. It has a direct impact on the enzymatic activity. If we would provide the enzyme with the optimum temperature, it would do the maximum catalytic activity. If the temperature would be beyond the optimum, enzyme would denature, and if the temperature is below the optimum value or is very kept very low, so ultimately the enzymes, they barely move, so they would not show any of the enzymatic activity. So simply for 2 marks, what you would require to be writing is that the temperature, it directly impacts the enzymatic reaction. And all the enzymes, they require an optimum pH where the enzyme would show the maximum catalytic activity. And other than that, like I have to be keeping the temperature as constant, because this is not something that I am investigating. Like if I were investigating the impact of the temperature, then the temperature was required to be changing. Now moving onward to the second part, and that was for the total of 2 marks, and they asked at that time that why the test tube 5 and the 6, they were included in the investigation. So I need to go back and I would have to make comparison of test tube 5 and the 6 with the rest of the four test tubes. Now, if you were to clearly see the test tube 5, it had to be compared with the test tube 1. And if you were to notice, like if we have the soya and if we have the urea, so we have the positive result. But if we have the soya and if we do not have the urea solution, then the result is negative. So this is basically an indication that the soya, if it releases the urease enzyme, then there has to be the presence of its substrate, then the result would be positive. But if we just have the soya beans, but we are not providing the substrate to the enzyme that would be released out of the soya, so the result that we obtain would be the negative results. And if you were to be looking on toward the test tube 6, so we have provided no beans and the urea solution was available, water was available, but yet we have got the negative results. So this is an indication that the beans actually have the enzymes being released out of them, and that is why when we have provided no beans, so ultimately no urease enzyme was available, and the urea solution would not be broken down into ammonia, and that is why we don't see any of the pH change. So simply you would say that the 5 and the 6 test tube that were basically included in the investigation to be acting as a test, and this is to indicate that the beans are the source of the enzyme, but alone the beans cannot change the pH until or unless we provide them with their respective substrate. So, how you were required to be writing the answer, you would say that the test tube 5 and the 6 act as a control, and the test tube 5 shows that urea is not broken down without the enzyme that has to be releasing out of the soya bean. As an alternative valid point, you would say that is test tube 6 that I have shown you above. So this would show that the beans alone are not the source of the pH change. There has to be the presence of beans, there has to be the presence of their substrate, and then the ammonia would be produced and that would change the pH. If we move forward for the 3 marks, they are asking that you have to state a conclusion that the student would make from the results of the test tube 1 to 4. So if we just simply revert back to 1 to 4, so you see soya is giving the positive result, and the positive result appear at 20 minutes. But if I talk about the mung, so we get no positive results. So this means that the mung are not producing any of the urease enzyme, but the soya is producing it. If I were to talk about the jack, so again I have got its result at an earliest time. So since a positive result is coming for the test tube 3, so I would say that the jack are also producing the urease enzyme, but their enzyme release is more comparative to soya because we are reaching to a positive result at an earliest time. If I were to talk about the broad beans again, they are not releasing any of the urease enzyme as we have not got any of the positive results. So the conclusions are that both the soya and the jack, they are producing the urease enzyme. This is 1 mark. Later I would make the second conclusion that the jack is releasing more of the urease enzyme because we are reaching the positive result at an earliest time. Here you would score 2 mark. And then you would say that the mung and the broad beans, they are not releasing any of the urease enzyme. So this is how you would score total of 3 marks. If I were just simply to read them up, so we would say that the soya and the jack has urease enzyme. Jack bean has more urease enzyme than soya because we have reached the result at an earlier time. And then the third conclusion would be that the mung and the broad beans, they are not releasing any urease enzyme out of them because we don't see any of the positive result. So this is how you would score total of 3 marks here. If I were to move further for question number 5, this appeared to in October, November of 2013. And they say is that the starch, glucose, and fructose, they are the carbohydrate. What I know is that starch is a polysaccharide, the glucose and the fructose, they are the monosaccharides. Further, they say is that fructose syrup is used as a sweetening agent as an alternative to sucrose. Like you see that we have to be using some sweetening agent in commercial purposes. So, as an alternative to sucrose, we are using this fructose syrup as a sweetening agent. Now, further, they are showing you a flow chart, and they say that this flow chart in figure 3.1 shows how the fructose is prepared from the maize starch. So, we have got the maize grains, and these grains they are crushed to be increasing their surface area, and their pH is adjusted. Now, out of the maize grains, the starch is released out, and then here you see that we are adding an enzyme 1 here. And when the enzyme would be acting on the starch, so we are getting a glucose syrup out of it. So the idea here is that the starch would be acted upon by the amylase enzyme, and that amylase is breaking down the starch into the glucose syrup. Now, since I have to be making the fructose, so fructose is an isomer of the glucose, and the glucose syrup can be converted into fructose syrup by the addition of the enzyme 2. So this means that before I cause the syrup, glucose syrup to be acted upon by the enzyme 2, I need to be eliminating this enzyme 1. Because if the enzyme 1 would yet be available within the glucose syrup, so ultimately this can impact the activity of the enzyme 2 that is coming here in between the conversion of the glucose syrup to the fructose syrup. Now they are saying that name the enzyme 1. So the enzyme 1 instance breaking down the starch to the glucose syrup. So this is amylase with us. Now moving onward, they say state why it is necessary to adjust the pH before an enzyme is added to the process. So the idea is very simple for 1 mark that pH is one of the major factor that have an impact on the enzymatic activity. Either you can write this or you could say that the enzymatic activity they require an optimum pH for their maximum catalytic activity. Or if you forget about this point, you can writing a third alternative point instead of first two. You would say that if there are extreme pH changes, so they can denature the enzyme. So this is why we are adjusting the pH. Moving onward for 1 mark, they say that the maize grains they contain the protease enzyme. Like these maize grains that we have got, they have got another enzyme in it, and that enzyme is the protease enzyme. And they say with reference to the processes shown in figure 3.1 suggests why it is important that these enzymes do not contaminate the glucose syrup. So the idea is very simple, you see that what I see here is that this glucose syrup that we have, so it should not have any of the protease enzymes in it that are coming from the maize grains, because you see that this glucose syrup further has to be dealt or need to be treated by an enzyme too. So if the glucose syrup would have a contamination of the protease enzyme, so they might denature this enzyme too. And if this enzyme too would be denatured, then ultimately we cannot make the fructose syrup out of it. So for 1 mark, you would say that the protease enzyme, if they are not eliminated, so they can denature the enzyme too. So that is why we are required to be eliminating the protease enzyme out of the glucose syrup. Moving onward for 1 mark, they have asked that the formation of the fructose syrup from the glucose syrup is carried out at a temperature of 60 degrees centigrade. Like if I were to be making this fructose syrup out of the glucose syrup, so here we have kept the temperature of 60 degrees centigrade. They say suggests an important property of the enzyme too that allows it to be used at a temperature as high as 60 degrees centigrade. So the property that the enzyme 2 would be exhibiting that since we are providing very high temperature of 60 degrees centigrade and still we are making fructose syrup, so this enzyme 2 has a higher tolerance to an increased temperature. So we would simply say that this enzyme does not get denatured at a high temperature of 60 degrees centigrade. Now, moving onward to question number 6, this appeared in the May-June of 2014, and this says that one of the component of the human milk is the enzyme lysozyme that is present in many body fluids and is responsible for breaking down the cell wall of the bacteria. So the human milk and many of our body fluids like the tear, they have the enzyme lysozyme in it, and that lysozyme have an ability to be binding or breaking down the cell wall of the bacteria and the bacteria would die. For the question number 6, I have included the D part only, and they say define the term enzyme for the total of 2 mark. And we would say that the enzymes, they are the biological catalyst and they speed up the rate of the chemical reaction. As an alternative point, you could also say that these enzymes, they lower the activation energy and then would speed up the rate of the chemical reaction. Now, for the second part that we have here, they have given you an investigation that they took the human lysozyme and they tested it on two species of the bacteria A and B, and they have also changed the pH. So this means that the pH is being changed. So this would be acting as our independent variable because we are having different values of the pH. They say the investigation was set up as shown in figure 2.1. The test tubes were kept at 37 degrees centigrade and the time was 24 hours. So if you were to see here, I have got the test tube number. This is 1, 2, 3, and 4. For the first three test tubes, we have in them the species of bacteria A, and for the fourth test tube, we have the bacterial species B. If we were to simply see the pH of the medium, so for the test tube 1 and 2, the pH kept was 4. For the test tube 3, this was 9. And again for the test tube 4, that has a bacteria B, so the pH was maintained at 4. The fresh lysozyme was added to test tube 1, this was added to test tube 3, and this was added to test tube 4. And you see, we are also including the boiled lysozyme for the test tube 2 as well. Like here, we didn't add any fresh lysozyme, but rather than the boiled lysozyme. Now, what the examiner or the investigator did next was that he would take the samples or solutions out of them, and then he would take them into the petri dishes and would see, and then I would add the bacterial culture into it and would see that whether the bacteria they survived here or they did not survive. Wherever the lysozyme would be able to kill the bacteria, so ultimately they would not grow in their respective petri dishes. Now, if I were to simply say later what they have asked that after 24 hours, the samples were taken from each test tube. Like these test tubes that we are given here, so they are taking the samples out of them, and these samples now they are putting them into the petri dishes, like the petri dishes that we have got, and then they incubated these dishes at 28 degrees centigrade for further 24 hours to allow any bacteria to grow. So if we just simply see, like we have added the lysozymes in all of these test tubes, with an exception from the test tube 2 where we added the boiled lysozyme. We changed the pH of them, and then after we have left them for some time, now we are transferring the culture from here to here in their respective petri dishes to see whether the bacteria has grown or no. If you were to look the key, so here for the first petri dish, no bacteria is growing. For the second, third, and fourth, the bacteria is growing. Now, moving onward to the respective questions, they say, explain the results shown in figure 2.2 by comparing the following pairs. For the 2 marks, we are required to be comparing the test tube 1 and 3. Now, if I just move back toward the test tube 1 that we have here and test tube 3 that we have here, we have the pH of the medium for test tube 1 as 4, and at 3 the pH is 9. We have added the same fresh lysozyme to both the test tubes. So our conclusion would be that the fresh lysozyme is able to kill the bacteria only at pH 4. But at pH 9 that we are given here, despite we added the fresh lysozyme, but the bacteria grew, so this means that the lysozyme was not active at a pH of 9. So how you were required to be writing the answer, you would say that the enzyme is active in pH 4 but not active at pH 9. And the bacterial cell wall was broken down in test tube 1. And ultimately, the bacteria did not grow in the petri dish 1, but it grew in the petri dish 3 that we have here. Moving onward, they want us to compare the test tube 1 and 4. So if we just revert back to test tube 1 and 4, for the test tube 1, the species is A, the pH is 4, the lysozyme is there, fresh lysozyme is there. For the test tube 4, the strain of the bacteria is B now. The pH is same, and we have got a tick here that the fresh lysozyme was added. But if we just compare the result, the bacteria B from the test tube 4 is growing, but the bacteria from the test tube 1 is not growing. So this is a clear indication that the lysozyme is successful at killing the bacteria A, but this enzyme is not able to be killing the bacteria B, so might be possible the bacteria B they are resistant against the fresh lysozyme, and that is why they were not being killed. So how you were required to be answering this for 2 marks, you would say that the enzyme was able to break down the cells of bacteria A and not B, and the bacteria B is resistant to the lysozyme. Or in other words, the other way could be that the lysozyme was not able to bind with the cell walls of the bacteria B, so that is why the B that we transferred from here to here is not being killed, but the A that we transferred from here to here, it got killed. Now, moving onward, they want us to make a comparison or to give him an explanation for the test tube 1 and 2. And if I were to move back, So you see that the test tube 1 and the 2 both have the bacteria A species. The pH is same, but the difference is that to the first test tube, we are adding the fresh lysozyme, but to the second one, we are adding the denatured lysozyme. And if we just compare with their petri dishes, so where there is a fresh lysozyme, the bacteria is not growing in their respective petri dishes, indicating that the fresh lysozyme at pH 4 is able to kill the species of the bacteria A, but once we would be using the boiled lysozyme, so that means that the bacteria A would now not be killed, and that is why when we will transfer them from their test tube to their respective petri dish, so this bacteria was able to grow now. So simply the answer would be that the boiled lysozyme is denatured in the test tube 2, and it was not active, the enzyme was not active to digest the bacteria. Or as an alternative point, you could say that this can also act as a control to indicate that the lysozyme is required to stop the bacterial growth of the species A. Now, moving onward to these 2 marks, they say is that the human milk also contains antibodies. Explain the benefits of the antibodies to a newborn child. So, when the newborn child is there, so its immune system is not that much active. So, if we would be injecting him with the antibodies, so the pathogens out of that newborn would be eliminated, and that child or the newborn child would not be getting any of the disease. And you may have learned it in the immunity chapter that the antibodies they provide the passive immunity. So for 2 marks, what you were required to be writing is that the antibodies that are going into the body of the newborn child from the human milk, they would stop the newborn child from getting disease. Or in other words, you can write as an alternative bullet point that these antibodies they would provide the passive immunity to this newborn child that we have with us. Now, moving onward to this question number 7, and this appeared in October November of 2014. They say microorganisms in the soil they release enzymes to digest the dead leaves. So wherever we have the dead leaves and they are lying on the soils, in the soil we have the microorganisms and they would be releasing the extracellular enzymes out of them, and that would break down the carbohydrate, protein, and the fats in the leaves to their respective constituents. For the total of 3 marks, what the examiner is asking is that explain how the enzymes they catalyze the chemical reaction. So we know it very well that the enzyme molecules they have the active site, and the shape of the active site is complementary to their respective substrate molecule. The substrate molecule binds with the enzyme's active site to be making an enzyme substrate complex, and later the enzyme molecule we know it would remain intact, but the substrate that got fixed into the enzyme active site, so it would be converted into its respective products. So simply how you were required to be writing it down that the enzyme has the active site whose shape is complementary to the substrate. The substrate would bind to the enzyme's active site and would result in making up of the enzyme substrate complex, and that later would be making the product, and the enzyme would remain unchanged or it would remain intact. If I were to move further, they say is that protease and cellulase, they are the two enzymes which are secreted by the soil microorganisms. Proteases digest protein, and they say suggest what part of the dead leaf cells are digested by the enzyme cellulase. So since the protease is digesting the protein, so the cellulase enzyme would be digesting the cell wall, and we know that the cell wall has this complex cellulose in it that would be broken down by this respective enzyme molecule. If I were to move further for the C part, and they say that we have presented you this table 6.1, and this shows the result of the study where we are comparing the decomposition of the dead leaves at two locations A and B. So if I were to just zoom in, this is table 6.1. We are given location A and we are given location B. And if we were to simply see, we have got the activity of the protease, we have that for the cellulase, and they have provided you with the soil pH, and they have provided you also the water content that we have in the soil. And if I were to compare the protease activity, so at location A and the location B, the protease activity is somehow similar. But there is a difference in between the activity of the cellulase. This is higher at location A, but this is lower at location B. Thus, if we talk about the soil pH, so we have got less acidic soil pH at location A, but the pH is slightly more acidic at location B. And if we just simply look in context of the water content in the soil, so the location B has higher water content, but the location A has a lower water content in it. So what they have to be asking is for the total of 3 marks that you are required to compare the enzymatic activity at location A with the enzymatic location activity at location B, and you would gain the credit if you are using the data from the table 6.1 to support your answer. Now we could simply say is that the activity of both the enzymes is higher at location A comparative to location B. The protease activity at location A and location B is somehow similar, but the cellulase activity at the location A is higher comparative to the location B, we say. So for 3 marks, the answer you were required to be writing was that the activity of the cellulase is higher at location A, and we have presented him the data that this is 4790, and we are also writing the unit which is micromole per minute than location B. Whereas for the location B, the cellulase activity is lower, and that is 2500 micromole per minute. The other point would be that the activity of the protease is nearly similar at both location, and all the enzymes they are more active at location A because we have got higher values here comparative to this. So this is how you would score total of 3 marks here. And now here they are asking is for the total of 3 marks that suggests possible reasons for any difference in the enzyme activity at location A than comparative to the location B, we say. Now, what we can make a conclusion here is that the location A and the location B they have the same composition of the protein in their dead leaves, so that is why the protease activity appears to be same. And since in the location A and location B the water content and the pH are different, but the protease enzyme is less impacted by it. But if we just look in context of the cellulase activity, might be possible that at location A, the cellulose content in the leaves were greater, so that is why the cellulase has a higher activity here and a lower activity at B. The other idea is that the activity of the cellulase is impacted by the pH. Might be possible that at location B, since the pH is more acidic, so that have partially denatured the cellulase enzyme, so that is why it is showing less activity at location B. So how you were to be putting it into the letters? So we would say that the cellulase is more active in lower moisture content, and the pH and the water content they do not have any impact on the protease enzyme because their activity, if you just see from the table, that this happens to be same at two locations. So it is not depending on either the pH or the soil water content. And other than that, we would say that since the cellulase activity was higher at A, so it might be possible the leaves A had got more cellulose in A than at B. So we would say that the amount of the cellulose present in the leaves varies at both locations. As an alternative point, you would say that the lower pH that we have got of 3.5 at location B, so this might be slightly denaturing the cellulase enzyme. So that is why it is showing the less activity at location B in context of the cellulase enzyme. So this is how you would score total of 3 marks here. Moving onward to question number 8 that we have here. This appeared in October, November of 2014. And what they have to tell us is that there are the students and they are investigating the activity of the enzyme by grinding some lattice leaves and then adding them to the solution of hydrogen peroxide. Now, the prior idea is that these lattice leaves that we have with us, so they have an enzyme in them, and the name of the enzyme is catalase enzyme. And when this catalase enzyme is going to work on the hydrogen peroxide, then two things would be formed out of it. One would be the water, and the other thing would be the bubbles of the oxygen gas. So here they say that the volume of the oxygen gas produced was measured. Now, since we are measuring the volume of the oxygen gas and anything that we measure during an investigation is given the name, and the name would go as the dependent variable, we say. And now the results of the students, they are shown in this figure 1.3, and what you are required to be understanding is that what comes on the x-axis. This is the time in seconds, and on toward the y-axis we are given the volume of the oxygen gas that is collected in term of the cm cube. Now, if I were to simply like zoom it more, so you see is that we have got total of five small boxes in between 0 and 1. So each small division is representative of 0.2 cm cube of the oxygen gas produced. And in terms of the description of the graph, what you observe is that as the time passes by, so the volume of the oxygen gas being produced is increasing, but until we have come toward the time of 90 seconds. Now, why I'm saying 90 seconds? Because here we have got total of 10 boxes. And in each of the small box that we are given over here, so this is representing again a time of 2 minutes. So 82, 84, 86, 88, and here we come to 90 seconds. So till 90 seconds, the volume of the oxygen gas increases, but from 90 seconds onward, this levels off. And at what point this is leveling off, if I were to come here, so this is 6.2 cm cube. If we will simply look on toward the gradient of the curve, so you see is that from 0 till 10 seconds that we have here, so the graph appears steeper. So here the volume of the oxygen gas being produced is 2.6 cm cube. Later again, the volume of the oxygen gas is being released, but this is being released at a lower rate. So these are some of the description point that you could go to your examiner. And now here he is asking for the total of 3 marks that you are supposed to be describing the results which are shown in figure 1.3, and you will gain credit if you use the data. So as I have said that as the time increases, so the volume of the oxygen gas increases until we have reached to the time of the 90 seconds, and then it would level off at a level of 6.2 cm cube. So you see I am like giving him the quantitative numbers. And then we would say that the volume of the oxygen gas, this is rapid for the initial 10 minutes, where the volume of the oxygen gas produced is 2.5, or you could say 2.6 cm cube. And then from 15 seconds, like I have got here as the 15 seconds, like this would be somewhere here. Then from 15 seconds, and then ultimately when we are going toward 80 seconds, so here the volume of the oxygen gas is being produced, but this is less rapid. If we move further for the 3 marks, they are asking is that explain the action of the enzyme during a reaction. So we have done like a lot of question at the top, and that enzyme has the shape of the active site, and this is complementary to the shape of the substrate. Both of them they would bind and they would make an enzyme substrate complex. That enzyme substrate complex would be broken down, and the enzyme molecule would come out as intact, and the substrate that we have got bonded into the enzyme's active site. So this would be broken down into the product. Now, since the substrate given at the top is hydrogen peroxide, so we have to be writing in that context. So what I would say that the shape of the substrate, which is hydrogen peroxide, this is complementary to the enzyme's active site that we have got in the extract of the leaves. The substrate is a lock, and the enzyme would act as a key. The substrate would bind with the enzyme's active site to be making enzyme substrate complex, and later from hydrogen peroxide, the product formed as oxygen gas. And afterward, the enzyme molecule active site would be available to be binding with another substrate particle. So I hope you would be clear with understanding the dimension of this topical past paper for the enzymes chapter from year 2010 and then coming till 2014. So see you in the next video where we would begin solving the past paper questions from the year 2015, and we would go till the end of 2025.